transcriptional factor in the cytoplasm, and that leaves the cytoplasm and it enters the nucleus. And when it enters the nucleus, it binds to our promoter region, and it can be an activator, i.e. it encourages RNA polymerase to bind and it initiates transcription, or it could be a repressor where the transcriptional factor binds to the promoter region, but then it blocks the RNA polymerase from binding to the promoter region as well. So you don't have mRNA being made, so it stops transcription. Let's think of a scenario. And the scenario is that we want to inhibit this gene from being expressed. And what does that mean? That means we want to stop this gene from to be made into a protein. So we don't want that protein to be made. But the problem is mRNA has already been made. So what can you do? Your mRNA has already been made, so we can't stop that, so it leaves the nucleus and it enters the cytoplasm. It's already been made, but we want to stop the protein from being made. So what can we do? The problem is we have mRNA already. So this is where we talk about RNAi. RNAi, we need to know what they are. These are very unique molecule. They are RNA, number one. They are non-coding RNA, so it does not code for any amino acids. Like your mRNA is an RNA and it codes for amino acids, the codons. But this RNA does not code for any amino acid. And the unique thing about this is that it's double-stranded. Generally, all other RNAs are single-stranded. And this is the only area where you'll find in your spec that this is a double-stranded RNA. RNAi is a generalized word. It's a very general word. Interfering RNA. There are two specific types you need to know for your exam. SI, short interfering, and that is only in animals only. There is nothing in plants, only in animals. And you have microRNA, miRNA, and that's found in animals and also in plants. So the unique thing about this is that it's found in plants. Looking at this video here, this is the same slide, but I added a few things. So what was our problem? Our problem was we have mRNA in the cytoplasm very close to be made into a protein, i.e. it's just about to start translation. But we don't want that to happen. This is the last step before protein being made. We don't want that protein to be made. So what we have over here RNAi. I'm being very general here. I'm not going to say if it's in plants or animal. This RNAi. It could be siRNA or it could be miRNA, any of them. RNAi is a double-stranded, and the RNAi basically it unzips and it becomes single strand. The other strand is degraded. The only one strand remains, and that strand is complementary to the mRNA. So it's going to bind to the mRNA in a complementary way. And when it binds to the mRNA is going to go into small fragments, and these small fragments, so it will be cut into small fragments, and that small fragment will then be degraded. So it's destroyed. So if you have mRNA being destroyed, can we make that protein? The answer is no. So what have we done? We have inhibited that gene from being expressed.